Hi guys, today we're going to talk about female gametophyte development. My name is Iwezu.
Marlene.
My name is Marlene Morales.
My name is Christine Saradine. So today we're going to talk about the developmental process and what the biological significance is of this process and how this process works at a mechanistic level. Female gametophytes, what are they? They are known as the embryo sac or megagametophyte. This is where the egg cell production and fertilization take place. They are critical to the plant by allowing pollen tube guidance, fertilization, seed development, and endosperm development.
The developmental process of female gametophytes occurs over two processes, the metagametogenesis and the megasporogenesis. In the megasporogenesis, it is the process of formation of the embryo sac. During megasporogenesis, a megaspore mother cell, which is a diploid, increases in size and divides twice to form 4 megaspores that are haploid. However, only one of the 4 megaspores are functional. It grows rapidly and forms the embryo sac containing one nucleus. Then, that nucleus divides into 2 daughter nuclei and moves toward the post of the cell, signifying the embryo. In the megagametogenesis, the functional megaspore produces a mature female gametophyte. That megaspore undergoes 2 rounds of mitosis without cytokinesis, resulting in a multinucleate coenzyte. Cell walls form around these nuclei, which then results in cellularized female gametophyte. Within the ovule contains 7 cells with 3 different types as a result of megagametogenesis.
So here we have the female gametophyte that develops within ovule to form the 3 antipodal cells, the 1 central cell, 2 synderid cells, and 1 egg cell shown in the photo above. The stages of megagametogenesis, we have the overview here so you can see what the names are of the OI, CH, AC, CC. The megaspore contains a single nucleus shown on stage FG1. Through 2 rounds of mitosis, the nucleus produces 4 nucleate coenocytes, 2 at each pole with large central vacuole in the middle, shown in stage FG4. In the 3rd mitosis, phragmoplasts and cell plates forms between the sister and non-sister nuclei, shown on stage FG5. A single spore at the proximal position becomes a functional megaspore, which becomes a mature female gametophyte that's inside the developed ovule on the left. During cellularization, 1 polar nuclei from each pole comes in the middle and then fuses to become a diploid.
So the biological significance has to do with pollen tube guidance, fertilization, initiation of seed development, and seed development. So the significance in a female gametophyte in an angiosperm reproductive system, it comes in many steps and processes. Mutant defects in the female gametophytes reveal regulatory genes for the function of endosperm development induction. There are 3 main steps for pollen tube guidance. One is pollen tube growth. This happens when the female gametophyte directs the pollen tube to the ovule. Synderid cells, these are guards that guard the entrance to the embryo sac in the ovule, which releases guidance signals that direct the pollen inside. Last step is releasing of male gametes. This is when fertilization happens. So in this TikTok, it's showing the petals, pistil, and stamen. You can't really see the picture. So the pollen goes into the stamen and goes down the pistil into the stamen. Mostly by bees. So this pollen tube guidance is important because without it, the female gametophyte is useless. The female gametophyte is the area in which the egg and central cell are located. The guidance of the pollen tubes are important because the pollen needs to be able to know where to go to be able to fertilize the egg and central cell. So pollen tube guidance in this picture, it is showing mutant embryo sacs. So embryo sacs are found to be the source of the attractant that guides the pollen tubes to the ovule.
Fertilization. This is a big step regarding female gametophyte because it gives its purpose. Fertilization is when the pollen has been transported to the embryo sac, which is the female gametophyte, and the sperm fuses with the egg to create the embryo, and a second sperm fuses with the central cell to create the endosperm. A syndicate cell death initiation happens after the arrival of the pollen tube in the female gametophyte. In normal fertilization, syndicate degeneration may be a physical process where the release of the pollen tube content into the synergic cell will physically rupture the synergic membranes. Or the synergic cell may be a physiological process induced by pollen via diffusible signal or through direct contact with the synergic cell.
So the mechanistic level of female gametophyte development, the mechanistic and genetic level of female gametophyte development plays a big role because all of the mutations that can occur change the phenotype of the plant.
I don't want to fall asleep.
And that TikTok is showed beautiful plants and the phenotypes. And mechanistic level connections between FG development and phosphorylase signaling were just suggested to have found that CKI1 mutant plants displayed FG lethality. Seeds could be obtained at a low frequency from a different allele, but were enlarged compared to wild type seeds. So on the right photo of cytokinin, we see the biosynthesis, degradation, and the function of AHK, AHP to give the response. CKI1 can induce a cytokinin response via the AHPs independently of AHK function. Gene mutations. A mutation in AGO9 gene. This results in the formation of ectopic MMCs in the ovule, and only the normally positioned MMC will form an embryo sac. Therefore, the primary function of AGO9 is in the siRNA-mediated transposon silencing pathway, and the mutated AGO9 mutants show active transcription of silent transposons. This is similar to transposon silencing by small RNAs in the germline of animals as well. So the mechanism by which AGO9 acts to restrict MMC fate is in the ovule is unknown, but however, mutants in other genes in the 24NT siRNA transposon silencing pathway, which includes RDRT, DCL3, and DNA-dependent RNA polymerase 4 and 5, have also shown to have the same effect. This suggests that the mechanism is directly related to the function of AGO9 and RNA-directed silencing and is not a separate function. These findings do point to the importance of RNA silencing for normal initiation of gametophytic development, and the mechanisms by which these processes are connected are important.
And lastly, we have some fun plants to look at and appreciate.
Someone needs to get down. I'm on notice, open digital run. You think I'm tepid, but I'm misdiagnosed. 'Cause I'm a stalker.